and it's a great pleasure, great honor to be here, and also to have met Dr. Bob Stairs, and also to give the Stairs lecture this evening. So hopefully I've got enough chemistry in here for this audience. So I guess to start off, I'm going to introduce the main player of the story and the organism that I've been working on or devoting the most of my time to over the last 20 years or so, and that's this bacterium or a genus of bacteria called Rhodococcus, and these are very widely distributed bacteria in the environment, so in soil and marine environments. They're one of the first bacteria that will grow up if you try and grow up bacteria from a handful of soil. And they can degrade an extremely wide range of organic compounds and have a very rich armamentarium, so a large number of many different types of catabolic enzymes, so enzymes that can break down different organic compounds. And so these bacteria have a lot of biocatalytic potential. And in fact, they are responsible for the most commercially successful microbial biotechnological application on the planet, and that is the production of thousands of tons of acrylamide each year. So there's some of these rhodococci have a nitrile hydratase that's utilized to synthesize the acrylamide from acrylonitrile. So just to give you a little bit more information about this organism or what makes it special, or one of the things that make it special is, I'm sure some of you, when you think about bacteria, some of you have heard of gram-positive bacteria and gram-negative bacteria. Well, this is neither one nor the other. So it's been classified as gram-positive bacteria, but in fact, and gram-positive bacteria do not have an outer membrane. So this is the inner membrane, and then the bacterial cell wall here. And gram-positives don't have an outer membrane. That's something that gram-negatives have, but this bacterium has an outer membrane. But what's special about this outer membrane is that it's got a very unusual fatty acid in it called a mycolic acid. And these mycolic acids can be up to 90 carbon atoms in length. So they are extremely long. And this is something characteristic that the bacterium shares with Mycobacteria. So I'll come back to that later. But this very special outer membrane makes these bacteria, both Rhodococci and Mycobacteria, resistant to chemical insults and antibiotics, actually. So we got into this organism through a genomic study when Genome Canada was just starting up back in 2000 or so, and this is when I just moved back to, or just moved to UBC to accept a position there as an associate professor. And our aim was to do a genomic study of these bacteria to understand and engineer their metabolic capabilities. And we chose this particular species of Rhodococcus called Rhodococcus joshii RHA1. I'm just going to call it RHA1 from here on out because two of us who were driving this project, myself and my colleague at the time, Bill Moan, were both interested in the degradation of PCBs because that was a big environmental pollutant. And this particular bacterium, which had been isolated by Masao Fukuda and Nagaoka, was extremely good at degrading PCBs. So we want to understand, first of all, why it was so good at degrading PCBs. So I was sort of like a biochemist interested in the enzymes that were responsible for this degradation, and Bill was a microbial ecologist. So we're coming at this problem from very different angles. And because neither of us knew a darn thing about genomics, we convinced our colleague Julian Davies to front for us. And so he came on board. And we had a wonderful collaboration with Masao as well in Japan. So before I sort of delve into the meat of this talk, I just want to show you the pathway that's responsible for the degradation of biphenyl, so why these organisms can grow on biphenyl, and also this pathway is used to degrade PCBs as well. You'll notice that this pathway has got two oxygenases in it. So these are enzymes that use dioxygen to affect chemical transformations. There's an initial ring hydroxylating dioxygenase that's an iron-containing enzyme, introduces dioxygen into one of the aromatic rings, both atoms of dioxygen, dehydrogenation, and then there's a second iron-containing dioxygenase that cleaves the ring at this position here. So it actually cleaves open the aromatic ring at this position, so we call it a meta. The resulting product is a meta cleavage product, or an MCP, and then there's this interesting hydrolase here that catalyzes the hydrolysis of this carbon-carbon bond here. And so it turns out, one of the reasons why I'm pointing this, taking the time to highlight this pathway to you, is because this is a very classic strategy for degrading aromatic compounds, and there's still a lot of work going on in my lab on degrading like lignin-derived compounds and things like that. And so this strategy is used in the degradation of a lot of these compounds. And as you see, it's going to come up again in the degradation of steroids. So, and I was keenly interested in these enzymes. Well, still am actually, but especially 20 years ago. And this actually, during my postdoc, I did a lot of study. I went to Germany just to study this enzyme here. And we were able to get the first crystal structure of one of these enzymes. I'm just showing you here. It's an iron-containing enzyme. You see in its coordination sphere, it coordinates two histidine residues and a glutamate. So you've got this thing called 2-histidine 1-carboxylate facial triad. And this turns out to be the first example of this kind of motif that's used in a wide variety of oxygenases, as it turns out, because there's 3 free coordination spheres in order to, that can coordinate the substrate and dioxygen in order to orchestrate a wide variety of oxidative chemistries. And in fact, we ended up getting a structure of the catecholic intermediate, the 2,3-dioxybiphenyl. You can see both hydroxyl groups coordinate to the iron, and that leaves space for the dioxygen to bind. And this binds as a monoanion in a bidentite fashion. And we've got additional spectroscopic evidence of that, and this has got important implications for the catalysis of the ring opening reaction, which proceeds. You see dioxygen binds, and then you get this bridged peroxo intermediate here between the iron and the organic substrate before you get ring opening happening. So, so that was kind of what was driving me in studying this bacterium ultimately. But I just like a little shout out to oxygenases before we move on, because they were discovered by Professor Hayaishi back in 1955, and it was actually one of these ring cleavage dioxygenases that he discovered. And so it turns out that oxygenases, or something to keep in mind, is that this is a second key metabolic role for dioxygen in addition to respiration. So the catabolism of many organic compounds actually occurs much faster aerobically for 2 reasons. First of all, because of aerobic respiration, which can support higher growth rates, but also because oxygenases can activate bonds that are otherwise very difficult to cleave. So I'm going to delve into now a little bit of genomics and some of the approaches that we use in my lab, and I wasn't too sure sort of, at what level I should provide background to this audience. So I thought I'd start with the central dogma of molecular biology, which sort of describes the flow of genetic information, right, from DNA, genes in the DNA, which are transcribed to RNA, messenger RNA, and then that's translated to proteins. And now we know this central dogma is being expanded upon a little bit because you can get reverse transcription of some viral genomes, for example, which are RNA. So, and then on top of this, I just want to point out, because we do a fair amount of metabolism, we're interested in the metabolites, that the proteins will, of course, catalyze a wide variety of chemical transformations in the cell that are organized into metabolic pathways. So another thing that we're really interested in, proteins, activity of the proteins give rise to a plethora of metabolites. Typically, a couple of thousand in the cell are present, different metabolites at any one time. So one of the reasons why I've given you this background is just so that we can talk about the genome, which is, of course, the organism's complete set of DNA instructions. The transcriptome, which is the collection of RNA transcripts that are present in the cell at a given time. And then the proteome, which is all of the collection of proteins that are present in the cell at a given time. And then the metabolome, which is, of course, the collection of the cell's metabolites that are present at a given time. And it's handy sometimes to think of these things in terms of the genome being an indication of what the cell is capable of doing. The transcriptome, so what transcripts are present, what genes are being transcribed or expressed at a given time, can be an expression of what the cell is preparing to do. The proteome is what the cell is trying to do. It's actually proteins in the cell, what proteins are present and their activities that are determining the phenotype of the cell, right? And then the metabolome is actually telling you what the cell is actually doing. And then, of course, there's areas of science that have sprung up to study each of these different phenomena. And so you got genomics, which basically relies on nucleotide sequencing of the genome of the cell, transcriptomics, also nucleotide sequencing, proteomics, trying to determine all the proteins that are present at a given time, largely based on liquid chromatography coupled mass spectrometry, or LCMS, and then metabolomics, which can typically be done, so surveying all the metabolites that are present in the cell and at what concentrations, also using LCMS or NMR, all right? So we make use of all these techniques in my lab. And then just before I delve into our pet organism, if you will, I'll just remind you that the human genome was actually not fully sequenced until a few years ago. It's still not completely understood, obviously. It comprises about 3 billion base pairs, right? Organized into 23 chromosomes. And those 3 billion base pairs code for or contain genes encoding about 20,000 different proteins. Okay? So compare that to a bacterium. So we're going to Escherichia coli, or E. coli, which is like the world's best studied organism. It's a gut bacterium. It's routinely used in the lab, even undergrad labs. So I'll just point out to you that the genome of E. coli is about 1/1000th the size of the human genome. But despite its much smaller size, and it's all on a single circular chromosome, but it still contains approximately a quarter of the number of genes that the human genome does. Okay? So in order for that to be true, all these genes are packed into the DNA a lot more tightly than they are in the human genome. Okay. And then, of course, once you've figured out, once you've sequenced the genome, you have to annotate the genome, and that means identifying the genes, which segments of DNA are coding for genes, and then assigning function to those genes. And sometimes functions can be assigned to genes through a process of comparing their sequences to those of known genes or characterized genes, and that involves an area field called bioinformatics. So by comparison of sequences, or they can be validated, the function of genes can be validated experimentally. I'll talk a little bit about how we do that as well. So, but underlying the bioinformatic predictions is the idea that proteins with similar sequences of amino acids or genes with similar sequences of nucleotides are evolutionary related. So they're called homologs, and also often have similar functions. Okay? And the other cool thing about bacteria is that genes that are involved in the same physiological process are often clustered together on the chromosome. So if something's involved in synthesizing an amino acid, for example, those 8 or 9 genes or 3 genes will often be clustered together. So you know they're all involved in the same process. In E. coli, I find this kind of interesting, even though it's the world's best studied organism, there's still over 1,000 genes, i.e. proteins, whose functions have not been precisely determined yet. So even in the best studied organisms, there's still a lot of unknowns. Okay, so where does this leave us with this RHA1 organism that we're studying? So the first thing we did was we sequenced its genome, and it turns out it's got a genome of about 10 megabases. So by bacterial standards, this is quite large, twice the size of E. coli genome. And this is because it's got a lot of different catabolic genes. And indeed, just based on sequence analysis, we predict that it's got a lot of oxygenases, over 200 of them, responsible for catabolizing all those different organic compounds I was alluding to earlier, and a lot of predicted aromatic degradation pathways. It also turns out that 65% of the genes from M. tuberculosis are conserved in RHA1. So even though RHA1 is this harmless environmental bacterium and MTB is this deadly pathogen, they've still got an awful lot of shared genes. So genes that do one thing in Mycobacterium tuberculosis do the same thing in Rhodococcus. So when we sequenced the genome of RHA1, we found the BPH pathway, as you might expect, because this organism was extremely good at degrading biphenyl, degrading PCBs. It turned out there were 3 homologs of this first enzyme, only one of them which was involved in biphenyl degradation, 2 others involved in degrading another kind of aromatic compound. There were 9 homologs of this ring cleavage enzyme, only 1 involved in biphenyl degradation, and a couple of others involved in alkylbenzene and alkyl-phenol catabolism. And then there were 5 homologs of this serine hydrolase that cleaves this MCP. So, and only one involved in biphenyl degradation, another in alkylbenzene. Okay, so that was cool. But when we published this genome, and this was back in 2007 now, and back then genome sequencing was not quite as easy as it is now. Now you can sequence a bacterial genome in an afternoon, or while you go have a cup of coffee or something. It costs almost nothing, but it took us over a year to sequence this genome. Anyways, because this was the first Rhodococcus genome that had been sequenced, researchers from all over the world who are working on different aspects of rhodococcal physiology started to contact us. And there was this guy in the Netherlands, Robert van der Geits. He asked us if our rhodococcus could grow on steroids. And that was like, oh, I don't know. So we test that. But before I get to that, I just want to talk a little bit about what steroids are, because there's some cool features about them. So these are I've got structures of a bunch of them up here. And so these are triterpenoids. They have a wide variety of biological functions. For starters, they're major constituents of eukaryotic membranes, constituting up to 45 mole% of some of these membranes. These membrane sterols vary depending if they're in plants, fungi, or animals. So animals have cholesterol, plants have these phytosterols, and fungi have this ergosterol. Their cholesterol is also converted to bile salts, which promote digestion and absorption of dietary fat in mammals. And then, of course, steroid hormones are important signaling molecules. And then there's a vast array of synthetic steroids, which are used for various pharmaceutical and agricultural applications. Now, the main reason why Robert was interested in the bacterial catabolism of steroids is because he was interested in engineering these pathways to convert steroids of low value, so cholesterol, for example, to synthetic steroids of higher value. And it turns out that while eukaryotes can synthesize all these sterols and steroids, they're completely incapable of breaking them down. They can convert them a little bit to hormones or bile acids or what have you, but they do not mineralize them. They cannot grow on them. They can't use them as a carbon and energy source. It turns out the only organisms out there that can do that are bacteria. And it turns out that the types of bacteria that are very good at degrading steroids are Rhodococcus and Mycobacteria. And these bacteria that have this unusual mycolic acid outer membrane. They're, of course, in the environment. They're responsible for decomposing biomass and these steroids in biomass and excreta. And yeah, as I've mentioned, Robert's interested in, his main interest was exploring the pharmaceutical or their biocatalytic potential here. So So yeah, he contacted us and asked us if we tested whether RHA1 could grow on cholesterol or other steroids. And so simple experiment, we did that. And sure enough, it grew like gangbusters on cholesterol. So that was kind of cool because now we had the genome sequence of this organism, first organism sequence that could grow on cholesterol and other steroids. So the first question we asked was, well, what genes are specifically expressed during growth on cholesterol? Because those are candidates for being responsible for the catabolism of the cholesterol. So we do a transcriptomic experiment in order to figure that out, right? Sequence, you isolate all the RNA that's in the cell while it's growing on steroids, and then, reverse transcribe the cDNA and then sequence the ensemble of transcripts, and then bingo. And it turned out there were about 80 genes that were highly upregulated during growth on cholesterol, and I'm sort of got them all here for you. And remember I told you that in bacteria, genes that are involved in one or a physiological process are often clustered together on the chromosome. Well, that's exactly what you're seeing here. All these genes here are clustered together on the chromosome, and I've just like color-coded them according to what their particular role we predicted they would have in the degradation of the cholesterol. So after we'd figured out what genes were involved in this process or were upregulated, expressed during growth on cholesterol, the next question is, well, what were the function of these genes? So in order to do that, we did a bioinformatics analysis, essentially, looking to see what kind of genes these things look like. And based on that and some biochemical work that had been done going back to the mid-60s, actually, we were able to figure out certain genes we predicted to be involved in the uptake, transport of cholesterol into the cell. Others were responsible for degradation of the side chain, which we predict would occur via a beta oxidation process. And then other genes that were involved in degrading some of the sterol rings. And what was kind of interesting in this analysis is that we saw that one of the rings during the degradation process was predicted to go aromatic, produce this phenol that got hydroxylated to produce one of these catacols, very similar to that 2,3-dihydroxybiphenyl that's produced in the degradation of biphenyl, and that got cleaved by a ring cleavage enzyme, and then that fragment was hydrolyzed by one of those serine hydrolases. So that was all in the genes that were up here that we had identified. So let's see. So this is all just a prediction, though, right? So now we have to validate the gene function somehow in order to really be confident that this is the process that's happening. So what I'm doing here is just focusing on 3 of the enzymes of the pathway, just to illustrate how we proceed here. So these are these 3 genes that, it's a set of 4 genes because this first enzyme, it's 3 enzymes. This one comprises 2 components. That's why there's 2 genes encoding it. But this is a monooxygenase that catalyzes the hydroxylation of this cholesterol intermediate to get this catechol. And then we've got this ring cleavage dioxygenase, cleaves that bond right there, just like in biphenyl degradation. And then you got this serine hydrolase that will cleave this bond right there, carbon-carbon bond. So if we're trying to validate the function of these genes, one thing we can do in bacteria is use special molecular biology tools that will allow us to knock out genes in a very precise way. Okay, so we can do this targeted gene deletion. Oh, sorry, I skipped ahead a bit here. So I just wanted to, this was just to emphasize the homology or the similarity between the biphenyl degrading enzymes that I'd worked on for years and years and these newly discovered steroid degrading enzymes. So this is the ring cleavage enzyme here, BPHC. It's a direct homologue of HSAC and this serine hydrolase here in both pathways. So this explains why, in part, why there were so many homologues of BPHC and BPHD in the RHA1 genome, right? Some of them are involved in steroid degradation. So this one here had been actually annotated originally as BPHC5, one of the 5, anyways. But there is a little link coming up later to that. So anyway, so we can knocked out the gene predicted to code for this ring cleavage enzyme, partly because that's a enzyme or this gene and enzyme is sort of a system I felt very comfortable with because I'd been studying it in another context. So we knock out the gene. So when we delete that piece of DNA in the middle of that gene, it means the gene's no longer functional. So the enzyme that's encoded by the disrupted gene is no longer functional. So essentially, we should be blocking the metabolic pathway at that step. So that now, if this mutant, where we've knocked out the gene, we should no longer be able to grow on cholesterol. But if we grow it up on another carbon source like glucose or pyruvate that it grows on normally, and the mutant will grow normally on that carbon source, we should be able to then incubate those cells with cholesterol. And if we're lucky, they will accumulate this metabolite in the culture media. And so that's exactly what we found. The mutant didn't grow on cholesterol, but if we added the gene back in a plasmid or something into the bacterial cell, process called complementation, then that mutant grew normally. So we hadn't inadvertently introduced a mutation somewhere else in the bacterium. And then when we analyzed the culture supernatant, when we incubated the mutant with cholesterol, we found that lo and behold, it was accumulating this metabolite, this catecholic metabolite that I'll just refer to as DHSA. So that was great, and we confirmed the structure of this metabolite by NMR because we were able to isolate a fair amount of it using some semi-primitive HPLC. And of course, that gave us enough compound as well to be able to use that as a substrate for this enzyme, which we then purified and demonstrated that this was an excellent substrate for the enzyme as well, doing steady state kinetics. And so that was, there's two separate approaches, molecular genetic and biochemical, to validate the function of this gene in cholesterol metabolism. And it turns out that some of these rhodococci are able to degrade multiple or different kinds of steroids. So our rhodococcus species here actually has 4 different clusters of steroid degradation genes. One of them is involved in cholesterol degradation, another's involved in bile acid degradation. Cluster 2 is involved in androstenedione and testosterone degradation. And you can see in each of these different gene clusters, you've got a homologue of the ring cleavage enzyme, just highlighted in red here. And also homologues of that hydroxylase as well in pink. So again, accounting for the large number of homologues of these genes in rhodococcus. Okay, so a little bit more bioinformatics here, because when we realized that BPHC5 and BPHD2 were the 2 homologues in RHA1 that were upregulated during growth on cholesterol, well, I remembered that the genome of Mycobacterium tuberculosis had just been published, and there were these 2 biphenyl degrading genes that were annotated in that genome, and that made no sense because Mycobacterium tuberculosis didn't degrade biphenyl, and it lacked all the other known biphenyl degrading enzymes. So we checked to see whether those genes were in fact cholesterol degrading genes, and looked for the other cholesterol degrading genes found in rhodococcus to see if they were also found in Mycobacterium tuberculosis. And what do you know? They're all there. About 80 of them. And so this led us to a bioinformatic prediction that Mycobacterium tuberculosis could grow on cholesterol, because up to that point, that was completely unknown. So there were about, but this, 80 genes in Mycobacterium tuberculosis, which has got like 4,000 genes in it. So that's like 2% of the genome of Mycobacterium tuberculosis devoted to degrading cholesterol. And nobody had recognized this, but it was kind of neat because some people had done random mutagenesis in Mycobacterium tuberculosis to figure out which genes are important for survival during growth in macrophages, for example. And it turned out that defects in some of these genes led to defects in growth in some of these infection models, but people didn't know what they did. So, well, that was kind of neat. So that led us to start studying the Mycobacterium tuberculosis. Then I had to go figure out some stuff about Mycobacterium tuberculosis because I didn't know anything at that point. So Some of you may know this already, but this bacterium was actually discovered back in 1882 by Robert Koch, and he actually used this to develop Koch's postulates, which is really designed to establish a causative relationship between a microorganism and a disease. So I won't walk you through these, but what really, again, just to drive home the point, this bacterium had been studied just about longer for over 100 years, over a century, and nobody had recognized that it was degrading cholesterol during infection. So, and I just, again, just to put this in a more global context, Mycobacterium tuberculosis, or TB in particular, has claimed more lives than any other infectious disease in human history, over 1 billion deaths, and it continues to claim over 1 million lives every year. And again, that number slowly growing again since the COVID pandemic, just because of all the lockdowns and redirection of resources that happened during all of that. So it's still like claiming about 1.5 million lives a year, with 10 million newly infected individuals worldwide. So it's still an urgent global threat, particularly because of the perniciousness of drug-resistant strains. The treatment of even drug-sensitive TB is very toxic, involves a combinatorial therapy of 4 different drugs. It's quite lengthy, 6 months. It's essentially a chemotherapy, which makes you feel very unwell. So unless people are closely monitored, they'll often not complete their treatment. And of course, that leads to more drug-resistant strains. And the treatment of drug-resistant TB is even more arduous, typically 2 years, and much harder on the body as well. And then there's co-infection with HIV as well. So there's an urgent need for new therapeutics. I'll just go over the infection cycle of MTB quickly so we can ultimately appreciate the role of cholesterol catabolism. But it's, there's direct human-to-human transmission through airborne droplets. Once the bacterium's inhaled by the lungs, inside the lungs, there's a particular kind of immune cell called alveolar macrophages, which will phagocytose the bacterium. And usually those macrophages will destroy invading microorganisms, and sometimes it can do that in the case of MTB, but very often what happens is the bacterium will arrest the phagocytotic process in the macrophage so that it can actually survive and multiply in the macrophage, in the phagosome. And as it does that, other immune cells, other macrophages are recruited to the site of infection, and then eventually a granuloma is formed with a lot of necrotic material in the middle of in the center of this granuloma, or tubercle as it's sometimes called. And the body, as long as the individual is healthy, can often maintain or wall off the bacterium in this state for decades sometimes. But then if the immune system breaks down, then this granuloma will break down, caseate, and bacterium will start replicating again and then be released and coughed up, and the infection cycle is reinitiated. So one of the things we wanted to do then was to see what the effect of cholesterol catabolism would be on this infection cycle. So to do that kind of experiment, you need an infection model, and infection models vary depending on the disease. It turns out that guinea pigs are a very good infection model for Mycobacterium tuberculosis because they form the same kinds of granulomas during infection that humans do, whereas mice do not do this, or many strains of mice don't. So we knocked out HSAC, again, that ring cleavage enzyme, in Mycobacterium tuberculosis, conducted an experiment with an aerosol infection of the guinea pigs. And you can see here that after 8 weeks, guinea pigs infected with the with the mutant have lower bacterial burdens in the lungs, and there's slower dissemination of the bacteria to other organs in the body because it is a systemic disease, to the spleen in this case. And you can see that there's less pathology in lungs of animals that were infected with the mutant. So again, fewer numbers of granulomas and smaller granulomas, and less pathology also based on the histopathology. And we also did these experiments in mice and found that it was attenuated in mice as well. But the important thing, though, is that we're not knocking out the disease altogether. So we're slowing down the bacterium in the guinea pig model, but it can still cause disease in that model. At this point, other research groups are working on this, and people have knocked out the transporter, other enzymes in the pathway, and in these systems as well. We saw attenuation in different infection models, mice in this case, but again, not knocking out this pathway didn't seem to eliminate the ability of the organism to survive intracellularly or in these models. Okay, just to come back to this infection cycle, though, I just want to point out that cholesterol is a major component of the phagosomal membrane. So during growth in the macrophage, there's lots of cholesterol that the bacterium has access to, and also at this later stage of infection, in this necrotic material in the granuloma is about 50% cholesterol. So there's lots of cholesterol present in the human host for the bacterium to utilize. Okay, so we went through and validated other steps of the pathway, essentially using the same methodologies that I described to you previously for HSAC, so knocking out genes, using mutants of Rhodococcus to produce metabolites for us to validate, for us to study the MTB enzymes, and using this sort of approach, we validated all of these, the functions of all of these different enzyme systems in red here. And other, Nicole Sampson down at Stony Brook validated the functions of a lot of these side chain degrading enzymes as well. So that left us with the question of, so we figured out the pathway largely to the degradation of this compound here, which comprises about 13 carbon atoms, so about half of the cholesterol molecule, which I'll just refer to as HIP. And it was completely unknown how HIP was being degraded. So that was sort of what we turned our attention to next. And we noticed that in all of these cholesterol degrading clusters, there was a cluster of genes of about 14 genes whose function had been completely, was completely unknown, and seemed to, these genes were conserved in all known steroid degrading bacteria, and many were predicted to be beta oxidation enzymes. So we hypothesized that these genes were responsible for the degradation of this HIP molecule, and that probably because they were beta oxidative enzymes, it probably involved some sort of degradation of some sort of acyl-CoA intermediate. So we noticed that when we looked at these genes, one of them was predicted to be an acyl-CoA synthetase, FAD-D3, so we knocked out that gene. And when we did that, we found that the mutant still grew on cholesterol, but only to about half the final growth yield as the wild type. So that's consistent with the mutant only being able to derive energy from half of the cholesterol molecule. And also when we looked at the culture medium, lo and behold, it was accumulating HIP. So we used that HIP, purified it, and then when we incubated the HIP with the purified FAD-D3 enzyme, ATP and CoA, we were getting the HIP-CoA being synthesized. So this led us down the trail of figuring out some of the HIP degradation pathway, and we started knocking out genes in this pathway. I'm not going to walk through all of them. Basically the same techniques I've been talking about so far, but a little bit of variation, because it turned out So we focused on IPDAB genes, these two genes here to begin with, because they'd been predicted to be essential for infection based on some mutagenesis studies that other researchers had done. And in fact, Robert had patented an IPDAB mutant as a live vaccine for Rhodococcus equi, which causes a pneumonia in foals. So when we knocked out these genes, we found that the mutant was unable to grow on cholesterol at all. So here's the wild type in black, and then the complement with the IPDAB genes added back to the mutant grew normally. But this mutant could grow normally on cholesterol. And the other interesting thing was this mutant was completely unable to grow inside of macrophages. So this seemed to be like very important in cholesterol degradation. When we looked in the culture medium, supernatant though, no metabolites were accumulating. And we suspected that this is because the metabolites that were accumulating were acyl-CoAs. And acyl-CoAs are not going to be secreted by the cell. So we're going to have to look at the intracellular metabolites now. So we had to develop some new techniques for extracting those CoAs. And when we did that, we could see that wild type bacteria were accumulating things like free CoA, acetyl-CoA, succinyl-CoA. But then in our mutants, in addition, they had these unique cholesterol-derived CoAs that were being accumulated. So we were able to isolate larger amounts of these acyl-CoAs by working with very large culture volumes, sometimes harvesting cells from 40 liters of culture and then breaking them open. So that enabled us to get enough material for highly accurate mass spec and NMR. And also we had a fantastic collaboration with Vic Sneakus at Queen's, who synthesized some of these molecules for us in order to validate their structures. And that was fantastic because then that also gave us enough material to do enzymatic studies as well. So in the end, we were able to demonstrate that IPDAB actually catalyzes a ring opening reaction. So actually a reverse, a retroaldyl condensation, and got a crystal structure. Yeah, that's all I was going to say there. So anyway, so same basic approaches, and we were slowly able to piece together this HIP degradation pathway, and we've since gone on to validate some of the intermediates that we predict to occur by getting NMR structures of some of these intermediates and demonstrating that there's other the other ring is opened by EK20 through a reverse Diekmann cyclization reaction, which is kind of neat. So, so this is kind of where we are now with our understanding of how these bacteria are catabolizing cholesterol. So I guess the next logical question is, can we somehow target this for novel therapeutics? And many antibiotics are discovered by screening compounds for the ability to inhibit growth of bacteria on plates or in liquid media. And that's fine and dandy, but very often those conditions are not replicating the conditions that the cells experience during infection, right? So it would be nice to be able to recreate the host environment. So at this point, my collaborator at Cornell, Brian Vandervan, he did a really cool experiment in order to try and identify intracellular inhibitors of mycobacterial growth. So basically what he did was he labeled Mycobacterium tuberculosis with a red fluorescence protein called M-cherry. So as the bacteria grow, they are naturally producing this M-cherry, and so you can detect that by fluorescence very easily in a high throughput manner. And so now he can actually screen for growth inside the macrophage. So even as the M-cherry, even as the bacterium is growing inside the macrophage, you get an increase of fluorescence. So this can be done in a microtiter plate, like in a 384 well plate, for example. So that makes it very high throughput. And basically, you're screening for if your compound doesn't inhibit growth intracellularly, you're going to get this nice, bright fluorescent signal, and if it does inhibit, you're not going to get any signal. You're going to get inhibition of that signal. And so this is just to demonstrate that using a common drug used to treat MTB, so isoniazide. You can see this is just like the DMSO control, increase in the M-cherry signal in the presence of isoniazide, though you get no increase in the signal. So now Brian screened a library of over 300,000 small molecules and at a concentration of 10 micromolar, and he got about just over 2,000 hits that resulted in 30 to 100% inhibition of growth inside the macrophage. So that was pretty clever. And then what he did was he rescreened some of those compounds. So he looked at which structural families they belong to and things. And so he rescreened about 1,400 of them. And again, inhibition of growth in the macrophage, so intracellular growth, but then also for growth in regular rich broth. And you see not all of them inhibit that growth in rich broth. Some of them inhibit growth both in the macrophage and in the rich broth. Some of them didn't inhibit growth on the rich broth at all, but when he specifically measured their ability to inhibit growth on cholesterol, it turned out that about 132 of them were specifically inhibiting the bacterium's ability to grow on cholesterol. So that was pretty neat. He did a few other experiments with C4 labeled cholesterol, just to demonstrate that in the presence of these inhibitory compounds, no labeled CO2 was being released, sort of to validate this blockage of the pathway that he was hypothesizing. And this actually implies that these two rings here aren't being cleaved. So at that point, because we had analyzed a bunch of these intermediates that accumulated in our different genetic mutants, Brian asked us if we'd be able to predict which enzyme of the pathway his inhibitors were acting on. So to do that, we grew up MTB on cholesterol, treated with these compounds, and then analyzed the culture medium to see what was accumulating. And in the case of these two compounds, we found that this phenolic intermediate was accumulating. And that, of course, is the substrate for this monooxygenase hydroxylase that produces the catechol that I've already talked about a couple of times this evening. And it turned out that a few years before this, we had already characterized this enzymatic step and had actually purified the enzyme, so had an assay for it and everything all set up. And we were able to show with our purified enzyme that these compounds specifically inhibited the enzyme with IC50s in the low micromolar range. Okay, so at this point, that's where the Mycobacterium tuberculosis work sits. I'd like to switch gears a little bit because there's another really pernicious mycobacterial pathogen out there, an opportunistic pathogen called Mycobacterium abscessus. And this is an opportunistic pathogen that afflicts people with poor lung function, including those with cystic fibrosis. So I'll just take a step back and talk a little bit about cystic fibrosis. Its prevalence in Canada is about 1 in every 4,000 births. The average lifespan of a cystic fibrosis patient is about 44 years. It's due to diseases caused by mutations in the CFTR protein, which is an anion channel found in epithelial cells, okay, that line our lungways and a lot of our ducts. So that when this channel has a mutation in it, the ion balance isn't maintained across the membrane, and then in these ducts in which that lined by these cells, the mucus layer loses a lot of its fluidity. So you get a very thick mucus layer, and especially in the lungs and the pancreas and other organs. And with this thick mucus layer, sort of thick dehydrated layer, sort of things like the cilia aren't able to function properly, so it's much more prone to bacterial infections. Initially, again, cystic fibrosis is a systemic disease. Initially, patients would die very early in life from malnutrition because they weren't able to secrete digestive enzymes, but that can be overcome by adding digestive enzymes to their food. But then they run into problems with things like lung function later in life. I also wanted to point out that it was a work done in Toronto that identified the mutations in CFTR as being the cause of this disease. Work done at sick kids by, so again, just to emphasize then, this thickening of secretions in the airways increases bacterial infection. The major bacterial pathogen in these in cystic fibrosis patients is Pseudomonas aeruginosa, but Mycobacterium abscessus has been increasing in prevalence recently from 3% to 23% over the last 20 years. And again, just like MTB, the infection is very pernicious and very difficult to clear. So in the case of the abscessus infection, the treatment regime is 2 years of antibiotics, and it's only successful in 30% of the cases. And if it's not successful, then the outcome of the patients is extremely poor. Okay, so because it's a mycobacterium, and the cholesterol catabolic pathway is just about ubiquitous in all known mycobacteria and rhococci, we had to look at the genome in Mycobacterium abscessus. And sure enough, there were genes that we predicted to be involved in degrading cholesterol in Mycobacterium abscessus. And then, but much to our surprise, well, we found that Mycobacterium abscessus was a little bit different than MTB because it also contained a second cluster of steroid degrading genes. Now, obviously, I'm not drawing out all the genes here. I'm just focusing on a limited number of them. But based on their sequence and so our bioinformatic analyses, we predicted that these genes, of the second cluster, would be involved in the degradation of testosterone and androstenedione. So we've got these two pathways. But one thing that was kind of cool, I kind of showed you this cluster of genes before, and you may notice that these two clusters are missing some things. They're a little bit short. They're missing, in particular, that gene that's involved in hydrolyzing the carbon-carbon bond in the ring cleavage product. So it turns out that even though there's these two separate pathways, neither of them contain this HSAD gene, but their Mycobacterium abscessus genome contains one HSAD gene far away from the other steroid degradation genes for some reason. But because there was only this one homolog in the genome, we predicted that it would be involved in degrading. Well, first of all, we predict that abscessus can grow on cholesterol and it can grow on androstenedione, and that the degradation of both of these steroids has to proceed through the single homolog of HSAD. So to test that hypothesis, we knocked out this gene. But again, that's just bioinformatic prediction. So now we're going to validate that. So what I'm showing you here is the wild type abscessus growing on 4AD here, and in black here growing on cholesterol. And then when we knock out this HSAD gene, that's the red line here, the mutant grows on neither androstenedione nor cholesterol. Then we add back that wild type gene to the mutant. It restores growth on each of these two steroids. So it looks like both pathways are functional in this organism, and the degradation proceeds through this single homolog as we predicted. What's kind of interesting here, though, is that we didn't actually just grow on cholesterol, but actually on a mixture of cholesterol and acetate. So usually, a lot of our mutants, when we knock out a gene in a cholesterol catabolic pathway like HSAC or something else, the organism may not grow on cholesterol, but it'll grow on a mixture of cholesterol and something else. And here, it's not growing on the acetate, even though it's present. So that suggests that by knocking out the HSAD gene in this organism, a toxic metabolite is accumulating that's preventing the growth of the organism on this other growth substrate. So that's kind of an interesting observation. Okay, the other thing is that this mutant did not grow in an infection model, a model of macrophages. So here, wild type growing, and the mutant here in red not growing in the macrophage. Okay, so this is looking pretty promising as being important for infection. So I think that comes to the end of my lecture. So I'll just like to conclude by stating that steroid catabolism evolved in environmental bacteria. Cholesterol catabolism in particular is almost ubiquitous in mycolic acid producing bacteria. The final steps of catabolism have yet to be elucidated, so that's ongoing work in my lab. Intracellular pathogenic mycobacteria have adapted the cholesterol catabolic pathway to access an important nutrient during infection. The precise role in infection is still unclear. And finally, cholesterol catabolism is the potential target for novel therapeutics, actually even in Mycobacterium tuberculosis, because some of these inhibitors of intracellular growth can act on specific enzymes, but it turns out, and something I didn't talk about today, is that some of them actually act on regulatory processes as well. And so there's one of those actually in clinical trials to treat TB right now. So I guess I should finish up by just thanking the members of my lab who've done this work. For those of you who know Carmen Chung, there she is right there. This is at our recent Halloween party. These are different people working on cholesterol catabolism and also lignin catabolism in my lab. And I've been able to benefit from some amazing collaborations, both at UBC, people like Natalie Stranadka, who's helped us obtain crystal structures of many of these enzymes, Brian Vandervan at Cornell, Vic Sneakus at Queens was a fantastic collaborator, and now Laurent at in Montpellier, and also I thank CIHR and Cystic Fibrosis Canada and the BC Lung Association for funding for this research. So with that, thank you very much for your attention, and I'll be happy to answer any questions.